I thought I would move up a little bit from the subcellular scale. We'll talk a little bit about the subcellular scale, but also to larger scales. It's a bit of a broad title. I'll tell you what I mean by this. But before I start, I wanted to really mention the people that actually did the work, because it's a little bit of a heroic work. I haven't really talked about it, so this is also unpublished, and it's the first time I'm probably talking about it. And this is the heroic work of two experimentalists, Adrian and Julia, and the theorist who's shared with Frank Jülicher between us. And it's heroic because it's on a new model system that we haven't worked with before. So I get motivated by movies like this one. This is 6 hours in the life, about 6 hours in the life of the early zebrafish embryo. You see cell nuclei labeled, according to kind of height in different colors. And what you can see is that basically in just a couple of hours, a bag of cells is able to structure itself into anything that makes up a living organism. You can see the backbone here, the eye, lobes, the head structures, and so forth. So if I look at this as a physicist, this is obviously a very interesting type of material that can structure itself in ways that I'm just going to say, still don't understand much about. So what are the laws by which this material structures itself? So I think this is a reasonable question to ask. What are the physical laws that underlie structure formation in these types of systems in living matter? And this is kind of borrowed from what is life, and surely is one of the first who really asked this question here. So we attack these types of questions in two model organisms. The main model organism we work, so A, we combine theory and experiment, and I'll try to take you through this a little bit. And B, we've been mainly working, and this is our lab logo. We've been mainly working with a nematode worm. So I call this the hydrogen atom of cell biophysics. We can discuss that, but it's a prominent model organism in biology. It's very stereotypic in its development, and it's easy enough to handle that also the experimental physicists will have to work with it and like that. But the system we've now bridged to is the quail. I'll tell you why we've chosen to start to work with the quail in a couple of minutes. But of course, quail is a little bit like chicken, and there's a couple of things specific about birds. So now I'll show you quail development. This is a collaboration with the Gross Lab, in Paris, and also here over the course of just a couple of hours, everything you see the head form later again. This I've learned a lot about in the last 3 years about the Sunday morning breakfast egg.
You got quail eggs for breakfast.
Yeah, they're basically they're a little smaller. But so first of all, of course, the embryo develops on the yolk. So the little black dot you sometimes see, that's cardiovascular system intact. Okay. So anyway, I don't want to ruin your stomach breakfast. So, but the nice thing here, this is basically 2D development. It grows on the yellow. In essentially 2 dimensions. So, and this is, we'll get to more to this. Okay. So, of course, it starts to form 3D structures as it proceeds, but the early steps are essentially 2D. Okay. So, I'm going to make a statement that is oversimplistic, but I'm still going to make it. There's essentially one molecule that drives the mechanics that you're watching here, and it's myosin. It's the same myosin, or a slightly different version of myosin that moves your muscles. Yeah, it's a non-processive myosin. So I'm going to make it even more simplistic. It's not the one that I'm showing you here, but here's one of my favorite movies of all times. What you're seeing here is a processive myosin motor walking along its track, which is the actin filament. So think about this type of activity amplified a billion-fold, orchestrated, of course, appropriately. This is what drives deformations, shears, restructuring. Larger scales in living systems. Okay. And this doesn't happen on its own, it happens in a particular organelle of cellular structure. This is the so-called actin-myosin cortex. This is the structure that separates the plasma membrane from the cytosol. It's a thin structure, just maybe 100 nanometers thick, and it's mainly consisting of these tracks, these actin filaments, polymeric molecules, And the motor proteins that I just alluded to, the myosins that assemble into myosin unit filaments, and in particular non-muscle myosin too. So this this is responsible for any type of shape change of the cell. If a cell divides, it has to depart this spherical shape. This is generated, these types of shape changes are generated by active myosin. We look into this is from C. elegans. This is a recording from the one-cell stage C. elegans embryo we've done with the best microscopes that we have. You can see essentially everything. You see the tracks in red, individual filaments, almost resolvable or filament bundles. And you see the individual green dots are single such myosin unit filaments. So these structures are motors. They burn ATP. They generate forces at molecular scale. You can see that the structure is slowly flowing. These are just a couple of seconds in the life of the one. So a fair question to ask is, what does this structure really do? Or what type of structure is this? If I zoom out a little bit, and try to think about the mesoscale. So this brings us, of course, to force balances, atomizing physics, you might call it. And I'm just going to make the point that it is useful to try to understand what this structure, how this structure reshapes itself at a more coarse-grained level. There's two things to know. First, there's a huge amount of turnover in the system. I'm just showing you about 3, 4, 5 seconds here, but if you wait 30 seconds, all the filaments are gone and are replaced with new ones. 30 seconds to a minute. Same is true for the green dots, for these myosin motor proteins. So over a minute time scale, elastic stresses will relax. So this is really a fluid-like material, and of course, it's got a motor protein present that can burn ATP to set itself into motion. So this falls into the realm of active matter physics. This is essentially a two-dimensional active field. So here, perhaps a definition of active matter physics. Might not be needed for this audience, but I'm still going to read it out. So this is essentially a material that is at molecular scales driven out of equilibrium, precisely because it's got ATP, it's got chemical fuel available to generate forces at molecular scales that can turn to stresses at more mesoscopic levels. And the point I'd like to make here is that that's great for us to understand, but it really gives us a formalism to try to think about biological systems, because in many cases we don't even know what the state variables are in biology. We have many problems where we don't even know what the parameter state variable is. Well, at least here we have formalism that's, you can say, systematic to give us state variables and relationships between them. And what I'm going to tell you is just simple ways of trying to use this approach to make sense of a couple of biological questions. So this is really, for me, a formalism for describing biological matter. And over the years, we've been working for many years with Frank Günicher at the Max Planck Institute for the Physics of Complex Systems in Dresden. We've been really trying to decipher what types of activities of the cortex at the mesoscale are relevant for understanding cell shape changes, cell atomizing cortical flows, we'll get to this, and so forth. So the cortex can generate active stresses. In 2D, then they turn into active tensions. At a mesoscale that are isotropic and equal in all directions. And this can give rise to flows. Active myosin cortex can also, the filaments can align with respect to each other. And the structure can generate stresses that are larger in the direction of alignment as compared to orthogonal directions. So this gives rise to anisotropic active tension. This is important, for example, for cytokinesis. The cell generates a cytokinetic ring that can contract. And then we've worked a lot on a third activity which we've discovered, which is that the cortex can also generate torques. There's a surface normal vector, symmetries are appropriately broken. So this structure can also set itself into rotatory motion. And this is in fact what I'll talk about for the rest of today. The framework then is that of an active chiral fluid, where essentially you have a 2D fluid with a density of active torque dipoles. There's angular moment exchange with the substrate. The torque dipole, of course, again burns the fuel. So in this case, you can't portion away, torque balances in fluids. So you really have to take torque balances into account. So we work a lot with Frank to also understand the hydrodynamics of active chiral fluids per se and in relation to how they apply particular biological transitions. So why is this interesting from a biological point of view? Well, of course, if you go to larger scales, this immediately might bear into left-right symmetry breaking. This is exactly, what I want to talk about. At large, we are, from the outside, we are basically bilaterally symmetric. But that's true only from the outside. I'm going to show you a bird here because we're going to work with whales. So inside, we're, of course, asymmetric. The heart's always on the left side, which means that if we take a mirror image, we're not going to do the same. Okay? So here's the appropriate image. Completeness. Okay, so of course this is about body axis establishment, one axis after the other. This is usually how organisms develop. You first choose head to tail axis, then you choose, it can be spontaneous symmetry breaking. You just choose an orthogonal axis. Okay, this is not really a choice. Anything orthogonal can define your next axis, which would be then the front-back axis. But now, of course, you've got a problem or a choice to be made, because now you've defined a plane. Now you can put the heart either to the right side of that plane or the left side of it. This is a real choice to be made here. And that choice is always made consistently, and one can discuss all of years why that is such a consistent choice in evolution. But nonetheless, there is a consistent choice, so it's a reasonable question to ask, well, how do organisms in early development make this consistent choice? So how do they choose always the same axis? And of course, again, this is a decade-old problem that has ignited the minds, inspired the minds of biologists and physicists alike. There has to be a kind of underlying molecular chirality. Lewis Walpert called it an F molecule. I'm going to call it an F activity, some process that is left-right asymmetric on some scale, inherently that can then be kind of transferred to more large scale left-right asymmetric arrangements. And in fact, in higher eukaryotes like us and mice and so forth, this has been famously pointed to arise, shown to arise from the cilia. Cilia are kind of flagellar structures that point out of cells that have a handed beat. And that handed beat in complicated ways can then be amplified in order to establish left-right body axis in a consistent way. But this is not so in all organisms. C. elegans, the nematode worm, does not have these cilia, these types of cells at the times that this happens. And even birds, although they're not so far away from us, I'm going to say evolutionary, they don't have this kind of structure with the cilia that's also transport mode. So this is why we started to work with birds, because we started to investigate in the nematode how this might work. But now this gave us some inspiration in terms of active chiral fluids. But the next question really was on birds. And now we're just going to talk about these birds. Okay, so how do they establish the left-right body axis? Okay, so here's our birds at our institute, the first couple of batches a couple of years ago. Now here back to your Sunday morning egg. You have this patch. The embryo essentially develops in this patch on top of the yolk, as I've told you before. There's lots of interesting rearrangements. We're going to look at the movie again. But I just want to point you to kind of a converging flow that sets up a structure that's called a primitive streak. This is in the neural tube that closes later. And at the top of this primitive streak, there's a node. It's called Tuch's node. Okay, sorry. I'm ahead of myself. So first, it is almost 2D, but it's a multi-layered structure. Okay, so this convergent flow actually allows cells to form yet another tissue layer inside. So this is when the system really becomes three-dimensional. But before, you've got this so-called epiblast. That's essentially a double layer of cell, and the movements are essentially quasi-two-dimensional. Now, again, these are these flows. Here you see a primitive streak. And the important structure for us is going to be this node, this point-like structure here. Because around this node, this was shown by Jerome Gross. So first, okay, it's a 2D problem, as I've said. And I'm just going to make the connection to active fluids and perhaps active chiral fluids by stating that these are lots of cells. These are thousands, hundred thousands of cells. You don't even see the cells here. And they've got an actomized meshwork at the apical surface, which means this, and they're connected to each other, which means that this is really just one big contracting surface, if you want to think about it like this. But how do you orchestrate these types of movements? And in particular now, around the node, this is what Jerome showed, there's a rotation. Okay, so we track individual cells. So this is the first instance really of left-right symmetry break, left-right asymmetric morphogenesis. This rotation leads to a thickening of the tissue on one side, and then something happens we don't totally understand. This gets amplified, this cube, in order to really establish this right body axis. So here, we zoom in a little bit better, and now we see the individual cells almost. I might say, let's just play. This is a node. Just before it starts to retract, this is to help you. You can see there's a slight rotation here. There's a slight rotatory flow associated. You know, it turns almost into a little bit of a comma shape here. I'm just going to play that again. Okay? So just follow. Here's the node. Here's to help you. Okay, now you see this slight rotation. Okay. So how does this? So the rest, you know, we've been wondering, you know, what drives this rotation doing is what we're going to be talking about. How does this tissue rotation come about? Is it locally driven? Is it globally driven? You know, where and how are forces and what's generated to drive it? And what's the role of the node in this particular, you know, and perhaps even the geometry of the node driving this? Okay, so how, you know, this might also show you what you can do with living systems. So, you know, I really enjoyed all the talks this morning, and I was sitting there a lot of times just wondering, I wish we could do, you know, shear over 2 scales of 3, 4, 5 scales of time and length. Okay, so I just want to just put this into perspective. This is about as much as we can do at the moment with these types of systems. Okay, some people can do a little bit more, but you know, it's coming from a different end. Okay, so but what we can do is measure flow fields. And in fact, what I'm showing you here is a flow field which is in a co-moving frame of reference with that node. Okay, and we can do that over time. Okay, so time 0 would be the onset of node retraction, and you're not really going to see it much here, but there's a slight rotatory aspect to this, you know, particular at the onset of node retraction. This becomes a bit more apparent if we just decompose this flow field into symmetric, asymmetric, antisymmetric components. Okay, so here's the left-right symmetric component. Okay, there's some local flows, of course, there's convergence into the street mainly. And here's the left-right antisymmetric component, where you can see this prominent tissue rotation just around the node. Okay? So this is just decomposed. Now we ask, and this is playing it over time again. Okay, so you see that this rotation becomes stronger, and then you know, as time proceeds, it also vanishes. Okay? So how does node and streak orchestrate? How do these structures orchestrate these flows? So to kind of get a hand towards answering this question, we set out to maybe perhaps try to reconstruct this phenomenon in a physical model. And this physical model we're going to pursue in the you know, one might in the context of what one might call crack physics or boundary physics. I'm going to illustrate this on the next slide. So we're going to look at a fluid. 2D fluid, for which we can measure boundary velocities on this circular region, but we're going to add an additional boundary, which is the street-like region. Okay, so we're just going to measure fluid flows around the boundary and along the street, and then we're going to ask, what do we need to do in order to, from the boundary flow, predict or you know, either say probably predict the flow field in the plane, or account for the flow field in the entire plane. Probably better. So we measure the boundary flows, and then we ask, what do we have to do in order to calculate the flow field in the entire region? And we're going to illustrate this with just a 1D version of this problem, but just keep in mind that we're going to do it, of course, in the entire 2D problem. So we measure the boundary velocities. We have fluid flow along the line. It's a 1D compressible fluid. This is going to fall into the realm of active matter again, and 1D active matter. And you know, we've spent much time on this, and it's well known. You know, in fact, so Meisen force generation gives rise to a contractile active stresses. Active stress gradients will drive fluid flow. There's a shear rate. This is a compressible in 1D, also compressible in 2D fluid. So there's stresses that arise from a rate of compression, and then there's an external friction coefficient because the whole thing, in fact, sits on the yoke, sits on the substrate, and there's the possibility of friction with respect to the substrate. Now the question is, do we need active tension gradients in 1D? Okay. Or, can we account for the flow field just by considering it to be a passive fluid? Or, are there gradients in active tension also required in order to calculate the flow fields in that entire plane? So that's the question we'll ask, and we'll pursue this. Now, this is what Jonas did in 2D. So here I'm going to show you in a couple of slides. They're always structured similarly. First, the boundary velocities along the some distance, about 300, 400 micrometers away from the node, and then along and around the streak-like structure. With the calculated flow field, here's the residual, the difference between calculated and measured flow field. So for time 0, which is the onset of node regression, this works. This is again a movie. I can show you that it usually works quite well. This is now looking at the left-right symmetric component of the flow field. Now we're going to do the same with the left-right, with the antisymmetric flow field. Okay. So now here are the antisymmetric components of the boundary flows. Here's the calculated antisymmetric flow field. Here's the residual between calculated and measured. Again, over time, seems to fit quite well. Towards the end, this is when this starts to depart from the prediction, but this is also the onset of neural tube closure, of setting up the structures that give rise to neural tube closure. So this is perhaps not totally surprising that it starts to fail at later times. If we don't include this node streak structure and just try to account for the flow fields by the circular boundary conditions, it completely fails. It's just an important point. So there's a lot that the node structure and the boundary flows at the node structure are key. So here's the entire flow field. Here's the calculated one without that node structure. So this is completely not captured. Okay, so that actually means left-right symmetry is broken at the epigraph boundaries, in particular at the streak and the node. Okay, so from this, how can we now infer back the forces and torques, you can say, that drive these flows? Well, this is rather straightforward because we just, with known viscosity, we can determine boundary stresses, and from boundary stresses, we just integrate on the boundary, we can extract the torque and the force. And here you can see that in essence, this node structure is generating the torque. So it's the node that's generating, presumably that's generating the torque to drive the torque flow. So finally, how are these torques now generated? We started to wonder. Okay, because you have the substrate, you have the yoke. So if you generate a torque, you're going to have to generate it with respect to something. So we started to wonder whether now the three-dimensional nature of the system does come into play a little bit. Okay. In particular, because if we've just focused on microscopes through the whole structure, and this is also reported in other, in the chick, there's a kind of an ingression associated with this primitive streak and node, and then there's a rather structure that has lots of ECM is the proper word. There's a real connection that seems to be to the other side of the tissue. So we wondered if this is kind of required to generate it withhold the counter torque in order to start rotating the tissue up here. So to test this, we just so here's the idea. So you have your epiblast tissue. It folds down. So maybe the geometry is more like this. So this is kind of continuously bending down. And maybe if you're now generating torques, here and on the sides, this would give rise to total rotation at the top side. But you're going to have to count, you need the substrate to inject angular momentum into the whole system. Okay. So is this the way that it works? Well, there's one way to test it, which is to remove the bond, to try to just remove that whole structure down there and see if you still get the rotations or not. Prediction would be that you should. So This is, in fact, what we did. This is a very delicate experiment because you're really taking apart this embryo, and we have to really make sure that it's still fine and happy enough for at least a couple of hours. And so we scrape off this whole layer, we've just done it a couple of times. I'm just going to show you that original movie again. This is what I showed you before. Normally we see this rotation. I think I wrote 6. I think by now we have 9 movies that show, here's the node. It's a bit harder to see here, but there's essentially no rotation associated with that movement. This again now is a situation where the entire bottom side has been scraped off. We check that they are happy and the cells still go into the 3rd dimension and so forth. But for me, this is pretty good evidence. We have to do more statistics, but at least a little bit of evidence that this is, in fact, maybe how it works. So to summarize here, we've asked these 3 questions. I think we can summarize them like this. We have good evidence that this is really driven by torque generation at the node, and perhaps, I'm just using the word perhaps because the statistics aren't quite sufficient yet, this node needs to be connected to the other side to be able to generate this counter torque. So how much more time do I have? So I'm just going to use probably 3 more minutes to go back to C elegans, because this system is great to study a complex orchestration of a major event in the early life of a bird. But a key question that how is this driven in terms of molecules? Which molecules are responsible now for torque generation? And here, the C elegans system, never one is a much better system to study molecular contributions, for example, to active torque generation. And this is because we've got all kinds of techniques that make it easy for us to remove proteins from the cell and then to see what happens. I'm just going to take you through one experiment that illustrates how one could go about. And that also shows us that torque generation is a complex, also molecularly, the orchestration in molecular terms is not quite as straightforward as one might think. So back to the worm. And I'm just reporting on work that's almost 10 years old now, where we've investigated. I showed you in red and green before a little subset of this one-cell embryo of the nematode worm. This was a more fancy microscope. This is a microscope with not quite this resolution, but what you can see here quite nicely is these cortical flows. So what you're looking at is a one-cell embryo. We're squishing it flat on a microscope slide, so we're just seeing the bottom surface of it, but there's a whole 3D structure behind. What I want you to take from this movie is that this cortex, this actin myosin cortex, you're looking at the myosin, the green molecules here, is flowing from this side to this side. This is, in fact, important to establish the head-to-tail body axis. But this looks like it's just flowing from this tip to this tip. That's not quite true. It's hard to see here, but if you quantify it, it becomes apparent. There's also a slight chiral aspect to this flow. This becomes amplified under particular perturbations, genetic perturbations that we can pursue. So here's a hyperchiral embryo. I guess you can see that it's rotating now. Where with Planck together, we spent many years, and Sebastian Flutter was the PhD student at the time, to come up with a kind of active chiral fluid, this force balance description of this phenomena, which is in fact capable of to look at where the myosin motor proteins are, the torque force and torque generating elements, and quantitatively relate the distributions of these motor proteins to the flow fields that are generated. This is the X component along the long axis, and Y component in green of this flow field. So this can be, with the thin film active chiral fluid description, we can bring this into quantitative agreement. Now, when you want to study how molecules contribute, you always have the problem that molecules often do quite a lot. If you take molecule X out, then there's no cortex. And then it's a little hard to study torque generation with cortex. Okay? So a trick we use is what's called a rescue experiment. And I just want to take you through this. So instead of starting off with a normal situation, we start off with a hyperchiral situation. Okay? This exact same scenario. And then we ask, which molecule do we have to tickle down in concentration just a little bit in order to come back to the situation where the flows are mainly on axis and not so strongly chiral? Okay? Because this has the advantage that you don't go to the full loss of functions that you want scenario. So we do this with a couple of candidates, and of course the first candidate was Meisen. Just the Meisen motors itself, because a key way to think about torque generation was that the Meisens just don't hit the pitch of this helical filament, and that will generate molecular torque, molecular torque dipole. But look at this. You reduce Meisen a little bit, it's flowing a lot slower because you have less motor proteins, but it's not really changing the chiral nature of this. Instead, the chiral nature of this gets completely removed when we get rid of a nucleator of the actin filament. So this is a molecule, it's called formin, that helps actin filaments to grow. It's not an ATPase, okay? It's using binding energies to propel itself forward, but it doesn't have ATP as an energy source, so it's more of a ratchet-like molecule, probably not very strong in terms of generating forces. But it's been shown before that this molecule will spin as the actin filament is grown. And if you get rid of this molecule, just a little bit, you see that we can recover the flows. Don't change velocity, but they change direction in some sense. Okay, so we lose the chiral aspect of the flows. So this is the last thing I really wanted to tell you. So these two molecules are cooperating in generating active torques. There are certain patch-like structures. I didn't really get to them. There's a way we can think about these patches to be important in order to generate active torques. But this is just hypothetical at the moment. Okay, so with this, I think I'll just stop, just leave you maybe with 3 thoughts. It's interesting to investigate living systems. I'm going to say mesoscale is relevant. We are, as a physicist, we're not going to be happy, I'm not going to be happy with an all-atom simulation of the cortex. I want to understand the force balance. That's at the mesoscale, and active physics gives us the formalism. And with that, I'll stop.